Hey kids, it's Mr. Willis. I'm sitting in the front row of the classroom on Sunday afternoon recording this for you so you can do well on your next test. Because some of you did bad on the last test that I just graded and I'll give back to you tomorrow. Why do I keep talking like this? So, y'all need to pay attention. There's only one test left. And then an exam, and for some of y'all, the prognosis is grim. So anyway, we're talking about development, animal development. And I was surprised, by the way, how bad y'all did on penis vagina. So development here, you need to pay attention, okay? This is chapter 42, page 796. And they're showing a sperm, that's what this is, invading an egg. That's what this big thing is. And all animals do this in sexual reproduction, and the sperm comes in, and it's got a little cap on the head of the sperm called the acrosome. See that word there? And the acrosome contains enzymes that'll eat through the membrane of the egg, you see. And it looks like just one sperm is releasing enzymes here, but really it takes a whole bunch of sperm to release enough enzymes to break through these two layers. There's an outer layer here called the corona radiata, and an inner layer here called the zona pellucida. And the sperm have to kind of break through both those layers before it can penetrate the egg and release the sperm nucleus, which it shows right here, into the egg. The sperm nucleus combines with the egg nucleus, and now you have a new nucleus with two sets of chromosomes and a new diploid cell, and that's what became you. A cell, this egg cell, is about the size of a period at the end of a sentence in your book. And after the sperm and egg come together, we now have what's called a zygote. Do y'all remember that? There's an interesting little thing that happens after the sperm penetrates the egg. There's a special kind of these little circular things here are called cortical granules, and they release an enzyme that goes around into this zona pellucida and forms a barrier that other cell can't penetrate. And so it only lets one sperm cell in the egg. If two sperm cells got into the egg, you'd have way too many chromosomes and the fertilized egg would die. So that's kind of interesting. It's called a cortical reaction that all of a sudden makes the membrane impermeable to other sperm. Isn't that interesting? So this is what was happening after your parents had sex. Ha! Go tell them you know. Go tell them tonight you know what happened. All that stuff about the stork they've been telling you, it's not true. There's a sperm up close, penetrating an egg. Here's how it happened on Family Guy. Wow. That was very dramatic. Okay, so you need to know the stages through which the cell goes in forming the early embryo. And so here it is. It's all on page 797. It shows it, top right of 797. It starts there with the zygote. And then the zygote starts to divide into 2 cells, then 4, then 8, 16, 32, 64. At this point where there's about 64 cells, it's called a morula. And then 128, 256. Yeah, I'm incredible at math. State Championship Math Team, 1986. Anyway, enough about me. Now we have a hollow ball of cells at about 256 cell stage, and we call that a blastula. So you had this ball of cells called a morula at about the 32 or 64 cell stage, and then it becomes hollow and it forms a blastula. All of you and I looked like this at one point in our lives. Do you remember? No, you don't. You were not paying attention then, as you are not doing now. Anywho, we call this stage where it goes from zygote to blastula, we call that cleavage, because the cells are cleaving in half. It doesn't have anything to do with the cleavage that you're thinking of. Y'all's mind is always in the gutter. Cleavage is a division, and so we're dividing the cell in half over and over. And I want you to know that this blastula stage, the cells are all about, the entire thing is about the same size as the zygote. So this blastula with 256 cells is almost the same size as the zygote with one cell. So what do you know that's happening during this cleavage stage? Think of a decent answer. Shout it out, someone. No one's shouting it out. What's wrong with y'all? It means the cells are getting smaller as we go through all these divisions. The cells are getting smaller. I'm sorry no one got that. How do I know a day in advance that no one got that? Well, y'all are pretty predictable, unfortunately. Anyway, the blastula, part of it starts to push inward. This gold area of cells that you see here, this side of this hollow ball of cells starts to push inward. It'd be like taking a flat basketball and pushing one side inward. And it becomes a little hat-like structure. It looks like a cell hat. And pretty soon it kind of curls around on itself, and we've now formed a structure that's called a gastrula. See, it says early gastrula? And it's basically two layers of cells. The outer layer of cells we're going to call ectoderm, and the inner layer of cells we're going to call endoderm. The endoderm layer of cells is going to become the digestive tube of the organism. And the outer layer of cells is going to become the skin and the nervous system. You need to memorize this. The ectoderm cells become the skin and the nervous system, and the endoderm cells become the digestive tube. And this little hole right here, that's called the blastopore, that little hole right there becomes either the mouth or the anus. Yes, I said anus. You can giggle all you want. Anus, anus, anus. But anyway, that hole is the mouth in a group of organisms that we call protostomes, and the anus in a group of organisms that we call deuterostomes. It's kind of funny, the anus, deuterostomes, you see? It's a good way to memorize it. Now, let me let you guess which one you are. Are you a protostome that formed mouth first, or are you a deuterostome that formed anus first? Go ahead and shout out your guess. Well, you're wrong. You're a deuterostome. You formed anus first. And so did I, by the way, and I'm damn proud of it. But anyway, that first hole that enters your body was your anus. And later, the mouth will form on the other side if you go later into the development of the organism. Later, you see down here where it says late gastrula, and they show the body of the organism lengthwise. A new layer of cells starts to form, this purple layer of cells. We call that mesoderm. And it forms from some of the endoderm cells. Some of the endoderm cells start to change and become what we call mesoderm cells. And the mesoderm is actually in between the endoderm and the ectoderm. And the mesoderm cells are going to become, whoops, sorry. What is going on? The mesoderm cells are going to become bones and muscles and the circulatory system and the kidneys and most of the organs that aren't the digestive system or the skin and nervous system. Those form from the mesoderm. So you need to get that straight. Endoderm on the inside, ectoderm on the outside, and mesoderm in between. And I'm not going to show you all these videos, but one of them's real good at showing what happens. I think it's this one. So watch this closely. Oh yeah, this is it. This is freaking awesome. Now this is a sea urchin egg, but a sea urchin develops the same way we do. I don't know if you know what a sea urchin is. It looks like kind of a spiky pincushion that lives in the water. It's related to a starfish.
Development of the sea urchin from a zygote shows the sequence in which most animal embryos develop. In the earliest stages of embryo development, continuous cell divisions in a zygote result in a hollow ball of cells called a blastula. With each cell division, the cells become smaller. As the embryo continues to grow, some of the cells of the blastula fold inward, forming the gastrula. All animal embryos, except sponges, pass through this gastrula stage. This opening becomes the mouth in protostomes, such as worms and insects, or the anus in deuterostomes, such as birds and mammals.
I'm touching its anus.
The third layer of cells, called the mesoderm, develops between the endoderm and ectoderm of the gastrula. In deuterostomes, the mesoderm is formed from clumps of cells that break off from the endoderm. The mesoderm gives rise to muscles, reproductive organs, and circulatory vessels.
So that shows it real well. See the mesoderm cells there? They broke off from this inner layer. Whoop, whoop, whoop. And those are the mesoderm cells. All those cells are just going to keep dividing, and they're going to fill this space in here, and they're going to become muscles and bones and circulatory system and kidneys and reproductive organs. All of y'all looked like this at one point. You were just about this stage is when this thing is embedded in the wall of the uterus. And this is going to grow into an organism, you see. In humans, it's embedded in the wall of the uterus. In sea urchins, it's just floating around in the water. And most of them get eaten by fish or something, come up and eat it. But some of them survive. So that's interesting. Here's a table showing the 3 layers. This is on page 2. Page 798. And the book talks about how lancelets are different from frogs, which are different from chickens. They all form a little bit differently. They have the same layers of cells, but they all go about their formation a little bit differently. One reason is because this yolk here, this yolk is food for the developing embryo. And chickens have a whole lot of yolk, a whole lot of food. But lancelets don't. So the development makes the yolk makes the cells look different. Yolk makes cells kind of reproduce a little bit slower. So you can read about that. Hopefully y'all read about, raise your hand if you've already read half of chapter 42 like I asked you to do. Go ahead and raise them. Deetra, count how many hands are up. None? Yeah. Thought so. And so this shows, this is a frog embryo, and that's all yolk right there, and this is the cells. And that's going to become that. This shows the formation of what we call the neural groove, the spinal cord. Remember I said the ectoderm forms the nervous system? So the blue cells here are ectoderm cells, and that's going to form the nervous system. The top of it here, which is called the neural plate, kind of forms a little groove. Then the cells circle around on themselves, and now we have this neural tube. That's the spinal cord. That runs all the way down the back of the animal. See, this is a cross section where the animal's been cut lengthwise so you can see down it. And that red thing there, that's called the notochord. That's going to form the backbone. So that's going to form the backbone, and that's going to form the spinal cord. And the backbone cells are going to wrap around the spinal cord cells to protect them. Unbelievable, isn't it? Now, I don't know if you've heard this word before, coelom. But a coelom is a body cavity. That's a word you should know. A space that forms in the middle of the body. That space there is going to get larger and larger. And it's going to become just an open space that's inside the body of the frog. And in that space, a lot of organs will grow. So there are most of the more advanced organisms are called coelomates because they all have a space. A lot of the less advanced organisms don't have a space. They're called acoelomates. And some of them have a little bit of a space. They're called pseudocoelomates. An example of an acoelomate is a flatworm, like a tapeworm that we studied. An example of a pseudocoelomate is a roundworm. But most of the organisms are coelomates. They have big spaces in their body where the organs fit. You're a coelomate. Pat yourself on the back. You're a coelomate. We don't need to watch any of that. Look at this picture. The coelom, this is at an even later stage, and look how large the coelom has become. That big space is the coelom. That's the gut. This is endoderm, and that's the tube where the food goes through. Comes in your mouth and comes out your butt and goes through that tube, and all the nutrients are absorbed, you see. Again, there's the notochord. That's going to be the backbone. And there's the neural tube. That's the spinal cord. Ectoderm is the outer layer. Mesoderm is the pink. That's the middle layer. Endoderm is the gold. That's the inner layer. Now, the next section of the reading, it starts on page 42.2. Discusses how cells become different. We call this process cell differentiation because not all cells are the same. Some cells become muscle cells, some cells become nerve cells, and so on. So the book goes into how do these cells become different, because when a cell divides, here we have a cell and it divides in two. If you remember the process of mitosis, this cell copies itself by mitosis. And so you go from 1 cell to 2 cells to 4 cells. Well, how do those cells become different? How can one group of cells become muscle cells and a different group of cells become nerve cells? When mitosis is a process that we learn that copies exactly. Well, mitosis doesn't copy exactly. Mitosis copies the nucleus exactly, and so you get 2 identical nuclei. But during mitosis, when the cell splits in half, which it has in this second picture here, the cell hasn't copied itself exactly. The cell just divides itself down the middle. And as you can see, the top cell is a little bit different from the bottom cell, not as far as the nucleus is concerned, but as far as the cytoplasm is concerned. On the bottom cell here, there are a lot of red dots in the cytoplasm. Those red dots are proteins. They represent proteins that were in the original cell. And they're calling these proteins maternal determinants. And these are proteins that were produced in the original zygote, in the original egg cell. And if you notice, we got a lot more red proteins, red determinants down here, and a lot more green determinants down here. So when we divide once and we divide again, these two cells on top have a lot more green determinants, and these two cells on the bottom have a lot more red determinants. The determinants help determine what the cells will become. So maybe if you got a lot of red determinants, you'll become the bottom half of the body. And maybe if you got a lot of green determinants, you'll become the top half of the body. And that's how the cells become different. That's one of the ways the cells become different. There's actually a lot of ways that cells can become different, but a lot of it relies on these proteins that are in the cytoplasm that we call determinants. In actuality, there aren't just red and green, which are two types of determinants. There's hundreds of types of determinants. And the cells will read these determinants, will somehow look at these determinants and decide what it should become, whether it should become a nerve cell or a muscle cell or a skin cell or a kidney cell or so forth. A lot of what decides what the cell becomes are these determinants. Are you getting this? This is highly important because it solves one of the mysteries. When a cell divides, it does not. It divides the nucleus evenly. The nuclei are almost identical copies of one another, except maybe for some mutations that might have happened during the copying process. But the cytoplasm is different. So those determinants are proteins that were found in the original egg, that were made for the original egg, and it helps determine what the cells will become. Now the cells can also communicate with each other by touch. And this cell is touching that cell. And imagine, let's go back a little bit. We can go back to this point right here. These cells right here are touching one another. And if this one becomes a muscle cell, then it can tell the ones around it to also become muscle cells through touch. And it's through proteins that are on their membrane that are touching one another can influence their development. So cells touching cells can influence one another, and that touch is important. And so this reading section, it goes on to show some experiments and such that were done. Talks about these determinants. Here's an interesting experiment that it shows. Here is a tadpole, a frog egg, and you see this, what we call this gray crescent, this moon-shaped gray crescent. If the gray crescent ends up, as this cell divides, if the gray crescent, if it divides where it splits the gray crescent in half, each cell will develop into a tadpole. Those are identical twin tadpoles, by the way. Tadpoles can have identical twins, kind of like us. If you divide the cell in half, where the gray crescent splits in two, you get two identical twin tadpoles. They're kind of like Stovall tadpoles. That one's Russell, and that one's Mac. Wait a sec, I messed that up. Russell and Lee. Hell, I can't tell the difference between them. Anyway, look at this. If the cell divides in a way that the gray crescent only ends up on one side, then only one side becomes a tadpole, and the other side becomes a bunch of undifferentiated cells that never differentiate. That means they all stay the same. So this is interesting. That means that the gray crescent has proteins that are required for differentiation to become a tadpole. The gray crescent are maternal determinants. This shows, this picture is not in your book, but it has something like it right here. This is page 803. This is a fruit fly egg. And this colored stuff are different proteins. There's proteins here, and different proteins here, and different proteins here, and different proteins there. And this is a fruit fly egg. And that will become the head, and that will become the tail. Here's some other proteins that have been fluorescently labeled. And do you see how they're in lines here? These lines become the different segments of the fruit fly's body. These proteins are determinants that are in the original egg that somehow those proteins tell that area to become the different segments of the fruit fly's body. And here's some different proteins, the way they're scattered around the egg. So these determinants are important in the development of organisms. This is kind of cool. It shows a fruit fly with 2 sets of wings. They found a type of gene called a Hox gene. And a Hox gene is a gene that controls large amounts of development. And in this case, the Hox gene was repeated. So the Hox gene is basically a gene that says, build wings. And there was a mutation here in this fly where there were 2 Hox genes that said, build wings. So the gene said, build wings, and we built wings, and we went through these long instructions for building wings, and again, another Hox gene that said, build wings again, and my gosh, you got 2 sets of wings on this fly. It's a mutant fly. We find these same Hox genes that control the building of wings. We find them in mice, and we find them in humans even. And a Hox gene is basically a set of instructions that say, go ahead and build this entire organ. And then there's the instructions for building the organ after the Hox gene. And if you read the book, it talks about how those Hox genes are important in building large structures. So if you ever hear on a test them talking about Hox genes, it's a gene it's like a go-ahead gene. It's a gene that says, go ahead and build the organ now. You can read about it in the book. Again, I got to cover this whole thing in one day, this whole chapter 42 in one day. So I can't stop and talk about everything for a long period of time. This is one of those one-day chapters where you have to do your best and struggle to get as much as you can from the chapter. Y'all don't do well on those chapters, but guess what? This is the last one. From here on in, we do every part of every chapter. Yay! So this last part is back to human development, which is on page 806. And it shows the egg being released there, the sperm penetrating the egg right about here, because the mommy and the daddy had sex, because they love each other and they want to make you. They're going to regret that decision. So the sperm and egg come together right there. And then you can see here we're going through the divisions. 4 cells, 8 cells, 16, 32, 64. Remember, we call this cleavage. And finally we get this hollow ball of cells. It's called a blastula. And that's going to implant in the uterus. This is what you all looked like about 7 days after fertilization. That's 7 days after your parents had sex. Shh. This is your mom's uterus. That's what it looks like. Isn't that gross? And then you implant on the wall of the uterus, and then you develop. Here's what you look like at 14 days old, a little disc of cells with a yolk sac that the disc is feeding off of. Yeah, you ate yolk at one time. You didn't have to eat it, you just absorbed it. There's the amniotic cavity that contains fluid that keeps these cells from drying out. Here's after 18 days, the embryo starts to form a little curved disc. After 21 days, the embryo gets even more curved. The yolk sac is starting to form a new membrane here called the allantois. That's where the wastes from the embryo are going to go. And that's going to eventually form the umbilical cord, a connection to the mom. 25 days after fertilization, you kind of a curved shape. Do you remember when you looked like that? Kind of looked like a little snake or something. And the allantois is forming this tube. The yolk sac starting to get used up. 35 days, that's 5 weeks. Now this is what you look like. And here's this umbilical cord forming. You're still stuck to the wall of the uterus. You're not very big at this point. You're barely visible. Maybe the size of the letter C in your book. If you look down at the letter C in your book, that's about how big this embryo is. Ha! That's what it looks like. That's what y'all looked like. Freaking lizard guy. Isn't that scary looking? That's a real picture with an electron microscope. Isn't that crazy? Look, you had a tail. All humans have tails at the start. The tail disappears. The tail is a vestigial structure that points to the idea that we evolved from organisms that had tails. Which is true. If you go back in our history long enough, you get monkeys and they have tails. Some of the different parts. Then later, a few weeks later, you start to look more like a person. You lose the tail. This is at 12 weeks. See, that last picture, that was 8 weeks, 12 weeks, 18 weeks. You're sitting there in your amniotic membrane that holds water around your body so that the cells can survive. The cells would die if they dried out, so they need water. 24 weeks. It's about 6 months. At this point, you could probably deliver prematurely and the baby would survive with medical help. You're upside down, ready to give birth. You're upside down because the head's heavy, you see. And the heavy head always goes to the bottom where gravity is. And this whole baby's going to come out the vagina. That must be painful. I won't have to worry about that. Oh, by the way, there's the umbilical cord right there, and it's connected to this organ. That's called the placenta. That's the feeding organ, where the baby's blood goes in and the mother's blood goes in, and the mother's blood and the baby's blood exchange nutrients. But this next picture shows that there's the baby's blood there, and there's the mother's blood there, and they never actually touch. There's a thin membrane separating baby's blood from mother's blood, and the nutrients can go across the membrane, but the blood never mixes. That's how come a mother with AIDS can give birth to a baby without AIDS, because the blood doesn't mix. Sometimes the blood will mix if there's a tear in the membrane, but usually that doesn't happen. The blood can also mix as the baby's being born, because the mother often bleeds. But it's interesting that the blood never really mixes. This is showing how you can take a sample. If you stick a needle in there, you can get a sample of the baby's cells that are floating around here inside the amniotic membrane. We call this amniocentesis. You may have heard of that. They stick a needle in there and they suck out some of the baby's cells and they look at them. Make sure the cells are okay. You know, if they saw too many chromosomes at this stage, they might know that baby's going to have Down syndrome, which is an extra chromosome number 23, if you remember. There's other ways they can get cells. They can stick a tube in the vagina and into the placenta and get cells from the placenta and look at them. They can do that at even a younger age, and they don't have to shove a needle through your stomach there. Instead, they stick a tube up the vagina. I don't know, maybe that's less comfortable. I wouldn't know. This is the kind of stuff Miss Brock's husband does, by the way. Mitch, he was in my class. They can even take eggs out of the ovary. That's what this is showing. Anyway, this stuff isn't from your book. This is from the old book. Anyway, so here comes the birth. So the baby has got to go out of the vagina and the water breaks. This little opening, it's called the cervix, and it's going to break open. Bam, you see it breaks open and all the water rushes out from around the baby. That's called the water breaking. And now that means the baby's coming. And this uterus, this whole muscular wall here, starts pushing out the baby. Push, push, push. And the baby's head is going to be pushed out. The head actually kind of expands, kind of stretches. See the stretched head? It looks like an alien baby coming out. And the baby comes on out, and then Mitch just grabs his head right there and yanks him out. And then after the baby comes out, they got to cut the umbilical cord, and then they got to pull the umbilical cord and the placenta out afterward. And then, like, animals will eat the placenta. I think Tom Cruise ate the placenta too. That's a Scientology thing. But most people just throw that away. I don't know what they do with it. You want to see video footage? Here it is. This is what you've all been waiting for.
Here's Dr. McFarlane in the delivery room. He's working with a mother who is well into the pushing and delivery stage of childbirth. In a few moments, we'll get to see the culmination of her 9-month miracle, the birth of her baby. This is the baby's head, emerging from the mother's vagina. That liquid you see is the remaining amniotic fluid being pushed out of her uterus. After Dr. McFarlane works the head free from the birth canal, the rest of the body is soon delivered. And now, mother and daughter share their first embrace. It's a very special moment to watch a new family meet each other for the first time. I just love happy endings.
Yay! Did you enjoy that? I have something neat I can do here with the beauty of computers. Whoop! I can make it go back in. See? It's disgusting. You ladies are gross. I'm kidding. We were all born that way. It's beautiful. Huh? Now, go out there and have a bunch of babies. Ladies. And guys, just, read magazines and stuff while they're doing that. Okay, talk to you later. Willis out.